So we are going to start going over some genetics and heredity information today. You can follow along with the information that we are going over on that note taker that was linked in Canvas. So genetics, plain and simple, the study of the way animals and plants pass on to their offspring. Things that are passed on by plants and animals to their offspring could include eye color, hair color, height, body build, blood type, intelligence, gender, among a variety of other things. The heredity are the characteristics that are passed on from both parents. So we have our heredity and our genetics. Then we have our cell. Some of this should be a review looking from your biology classes. But if we look at a cell, we know that that's the basic unit for all living matter. An adult has over 10 trillion cells in their body. Then inside that cell, we have the cytoplasm. This is the substance of the cell that surrounds that nucleus. That nucleus is the central point of the cell that has our genetic coding information. And this is what really dictates our growth and our reproduction. Our chromosome is inside of that nucleus. There are 46 chromosomes in each nucleus, and there are 23 pairs. So the chromosomes pair up inside of that nucleus. On that nucleus, we have bands of chromosomes that are known as our genes, and those genes contain our DNA. So if we look at this reverse, this is going to be like a building block. Starting really simple, we have our DNA. Our DNA is found on those genes. Those genes comprise a total of 46 different chromosomes in 23 pairs. Those chromosomes are found in the nucleus, which is inside of the cell, but it's surrounded by that cytoplasm. So if we're looking at a little bit of review from that previous slide, how many chromosomes are there in each cell? Well, there's 46 chromosomes, or those 23 pairs that we had talked about. So when we are looking at our reproductive cells or our germ cells, we are getting 23 chromosomes from the egg and 23 chromosomes from the sperm to give us those 46 combined chromosomes. Now, we do know of a genetic condition known as Down syndrome. So this Down syndrome shows us that we have an extra chromosome on the 21st pair. So that 21st has that extra chromosome, resulting in a total of 47 chromosomes, and that is the cause of Down syndrome. So looking at cell division, we are going to cover two separate types of cell division. We have mitosis and we have meiosis. So the first one is mitosis. This is when the cell divides by copying the DNA and splitting into a new cell with a normal number of chromosomes. So if we're looking at this, we can see that we have our DNA that's replicating. So I'm going from our one strand of our red chromosome and our one strand of blue chromosome to completing that pair here. Then what's going to happen in that mitosis phase is that those two chromosomes are going to split again to give us that one, and that whole cell is going to split off and give us two diploid cells, which will then go back here to repeat that entire process. So mitosis is the cell dividing and copying that DNA with a new number of normal chromosomes. This is our cell growth and our cell repair. Meiosis, on the other hand, creates half sets of chromosomes. So a woman would have 23 and a man would have 23. So from that female and from that male, we get those 23 combining to give us our 46. So if we're looking here, we have those half sets of chromosomes, and those are combining to give us those full sets of chromosomes. And then meiosis is going to take place in a couple different ways. So here we have those daughter nuclei that are splitting, and they're giving us almost a whole chromosome of the red with just a little bit of that blue, and almost that whole cell of blue with just that little bit of red, and vice versa on the bottom. Those are then going to split, giving us half sets of chromosomes again. So we have the half of the red and the half of the blue, creating those 46 pairs. This won't come up a whole lot. We just want to cover the basic portions of this so that you have a little bit of understanding regarding this. So now that we have a better understanding of how our cells are splitting and combining, we know that a female sex cell is known as an XX. A male sex cell is known as an XY. So in order to have a baby girl, we're going to get that X from mom, and we're going to get that X from dad, or that X from dad, and that's going to give us that XX, or a baby girl. Now, mom can only give us an X. We only have X's from mom, so we would get an X. Dad, if it's going to be a boy, is going to provide that Y. So here we have that X and that Y to combine and give us that XY for that baby boy. So this is known as conception. When the union of the ovum and the sperm take place, this is going to be conception. So if we look at it, gender is really going to be determined by the father, because the mother is only providing those X cells. So we need that Y to be provided to have a male gender. So the father is going to be the one that's determining that gender, unbeknownst to them. All right, so now we're just going to go through a little bit of vocabulary before we get into some of the Punnett square stuff. So a dominant gene is a more powerful gene that is seen in a person. So this is going to be a trait that we see in a person. It is more powerful in their genetic makeup. A recessive gene, on the other hand, is a weaker gene, and it kind of just hides in the background. It waits until it has another recessive gene to become apparent. So a recessive gene will not show up until we have two recessive genes. So when we are looking at this dominant gene and this recessive gene, this is where those Punnett squares that you have done in biology are going to come into play. So we have carriers. These carriers are going to have a recessive gene that is not visible. So they're showing these dominant genes, but they do have that recessive gene. And so we'll look at that in just a second. But those sex-linked are going to be, the mother passes down the X to the son that contains that recessive gene. And so the mom might not have it herself because it's recessive in her. But when she does pass that recessive gene on to the child, that child is going to show that recessive gene because it is a sex-linked or a X-linked trait. And we'll get into that in a little bit. But looking at those Punnett squares that you have on your note taker, if you look and you see that something like a capital B is brown eyes, which is our dominant gene, and a lowercase b is blue eyes, which is our recessive gene. If I have two dominant genes coming together, I'm just going to have a dominant. So I'm just going to have brown eyes. If I have a dominant and a recessive gene coming together, I'm going to have brown eyes, but I could carry that recessive blue eye gene. So if that recessive blue eye gene is seen in a later generation, that would mean that another recessive gene was provided. And then if you have two recessive genes that are provided, you're going to have that recessive trait. So that would be those blue eyes. All right, another important factor that we are going to look at with this chapter is multiple births. So multiple births, we're going to go through a couple different types, and then you're going to be breaking up and doing some research today. So multiple births, the zygote, this is the cell that is formed when the sperm and an egg are combined. So when the sperm and the egg create that zygote, it's going to be the cell that's formed. And this will make a little bit more sense when we get into talking about our twins. So we have that zygote. We have the cell of our egg and our sperm. Now mono means one. So if I'm looking at monozygotic, that is going to be identical twins. So there was one egg and there was one sperm. That fertilized egg, or that fertilized ovum, splits into two identical cells, which is always going to be the same gender. So they are identical. That cell just splits, causing one cell to split for identical twins. Now, di means two. So dizygotic would be fraternal twins. So we have two eggs that are fertilized by two different sperm. It's happening at the same time, but because there's two separate eggs and two separate sperm, they are going to be fraternal twins. So they will not be identical. They will look different. They might look the same, but they usually are differences. And then they will not be the same gender. If they are not identical, they will not have the same gender. And then the next type of twin that we have is a conjoined twin, also known as a Siamese twin. This is when the ovum splits apart, but the separation isn't quite completed. So at some point when that cell was splitting, it didn't completely separate. So the babies are going to be joined at some part of their body. So what 4 factors can contribute to multiple births? We have a couple different factors. One is a history. If you have a history of multiple births or twins, triplets, quadruplets, whatever it may be in your family, you yourself will have an increased risk of having multiple births in your reproduction. The second factor would be increased hormones naturally. So if you have any reason, whether it's a hormonal imbalance, whether it is going through puberty or even menopause, when you have increased hormones, you can release more than one egg during ovulation. So if more than one egg is released, there's the possibility to have more than one egg fertilized, which would result in multiple births. The third factor is fertility drugs or IVF were used. So with this, more than one egg is usually released just to increase the chances of conceiving. So more than one fertilized embryo would be introduced. This is going to be an IVF. So for fertility drugs, they're releasing more than one egg. For IVF, they're introducing more than one fertilized embryo. And that is really going to be seen with people that are struggling with infertility. So they are trying to increase the chances by releasing more than one egg or fertilizing more than one embryo and inserting.